So everyone, hello, welcome. We always record these meetings, but if you feel uncomfortable with like showing your face or when you ask questions, if you open your microphone, just let us know. We can definitely edit this later because all recordings are posted on YouTube. And wow, as I said before, saying again, welcome everyone to another Recon community meeting. Today we have here Daniel. He's a professor at the Víctor Alzamora Castro Graduate School, as well as director of the Laboratory of Single Molecules of the Faculty of Sciences at Universidad Peruana Cayetano Heredia. Prior to that, he did his PhD at the Catholic University in Belgium, where he gained a range of experiences in biophysics, biochemistry, and molecular biology. And he's here today to tell us a bit about his current work and ask for your thoughts on what would we want in a new and improved E. coli BL21 DE3 strain. Now I'll hand over to Daniel to share about his work to tell us what he's working on. So Daniel, the room is all yours.
Thank you, Sibel, and thank you for the invitation, Jenny, Yankee, for all the organization. I hope you enjoy this short talk. I don't want to make it very long, and I think many of us are interested in producing proteins regularly, and we always face some difficulties, and some of those we deal with them, but some of those maybe we can prevent them and improve our tools. I'm motivated in doing this from other side of another project. We were having, let's say, not only issues, but also ambitions to make things better for metal biosensors. We want to increase the signal so that the reporter really has to be produced greatly, but also producing a lot of reporter makes a high burden for the cell. So in this compromise between producing a lot of protein, but not harming the cell, many things can be, I think many problems come from this compromise between high production and high burden for the host bacteria, sorry. So when trying to develop something a little bit better for my development of metal biosensors, we come into a general problem on increasing the production of proteins without increasing too much the burden of the bacteria, for the bacteria. This common problem, normally you face it, you deal with it by tuning it down. So you can control as much expression as your bacteria can hold, can sustain. And then you go to lower temperatures or you decrease the induction. I mean, many things can be done. Yes, Ibel.
Yes, sorry to interrupt. I think you were planning to share screen, but we don't see the screen right now.
No, I'm still not.
Okay, great.
I'm still not sharing.
Perfect.
So, and thank you for this, because otherwise I will start talking too much. So with different motivations, I think we can wish to have bacteria to express recombinant proteins in a very tunable way. That was my main goal for starting this. I wanted something that is very tunable, and though it is very tunable, very finely tuning, I can express a lot of protein. That's what I want to achieve. And if this can be of general use, all the better. Now, and this is how I go to study a little bit more of how BL21 DE3 works and what can we do to make it a little bit better. And I have to say a little bit better because BL21 DE3 is already very good. I think probably most of us in the room have used it. So this title wants to motivate everybody into some of the things that we already want to improve. IPTG is quite expensive. And sometimes it breaks down in the lab. So it's not only expensive, but also not so stable as we would like it to be. And orthogonality is an issue for mainly the burden if you want to express a lot of protein, but also sometimes your protein will interfere with other in-house regulations. Not only your protein, but also the IPTG system is actually a system from carbohydrate metabolism. And if you add IPTG, you are also not only increasing the induction of your desired protein, but also for all the cyclic AMP system depending on CRP. So that is quite a lot to change in a culture, to add something that is inducing so many things. So why not to try with a transcription factor that comes from other places, from other bacteria? And then we can achieve a better tunability. Okay, a little bit of introduction about this burden problem. If we remember this DE3 system, we can have the T7 RNA polymerase. It is encoded in the chromosome of the BL21 bacteria, BL21 DE3 bacteria. And if we induce the expression of the T7 RNA polymerase, then this polymerase can transcribe a gene of interest that normally we introduce in bacteria just by a plasmid. So this gene of interest will be under the control of a promoter that is not recognized by E. coli RNA polymerase, but it can be recognized by the T7 RNA polymerase. This bacteriophage polymerase is very active, is a more or less simple protein. A difference with the E. coli protein is only a single peptide, is highly active, so it can transcribe a lot of protein from a single promoter. And this is how we get a lot of recombinant protein. And we, in my lab, quantify the expression of GFP in 3 different strains: BL21 DE3, BLR DE3, which is a derivative from BL21, and a K12 that has DE3 insert as well. The amount of fluorescence is depicted in green. Actually, the whole bar is the amount of fluorescence that we obtain. When you don't induce the expression with IPTG, you already have some baseline fluorescence, which is represented here by the blue column. When it is induced, you have, of course, a lot more. You see that there is some baseline here. We added some extra lac repressor to make it more stringent. It didn't improve much, and the total expression goes lower. In the case of BLR, you can induce the expression. The baseline is a little bit higher. In case you add repressor, you don't, a difference with MG, it actually expresses quite a lot. But the baseline was reduced a little bit. In the case of BL21, well, this was, you know, it has a high leaking, this system, but we didn't expect it to have such a high leaking with this plasmid. Without induction, we have a lot of fluorescence. And when we induced, there was a decrease in fluorescence. Normally, the expression plasmids have an overexpression of the lac repressor. So we added this overexpression of lac repressor, and then we go to a scenario much more similar to the other strains. So this is the BL21 DE3, and it's clear that in some circumstances, you can have a huge, huge baseline of production of your recombinant protein. In the moment of maximum growth rate, the burden can be quantified very easily. The maximum growth rate, when it is not induced, it is higher, and when it is induced, is lower. The case of MG, this was very clear. The case of BLR, the burden was very low. The case of BL21, there is really no burden that we can quantify in this case. Okay, this is another way to quantify the burden due to the expression of the recombinant protein. By adding more and more IPTG, you can increase the amount of GFP that is being produced for any recombinant protein, GFP for easiness. But of course, the more you produce of your recombinant protein, the lower your rate of growth. This slope, in the case of BLR, is very, very steep, which means that it's actually a very low burden. It's very tolerant to the burden of expressing the recombinant protein. BL21 turned out to be a little bit less tolerant to this burden, and K12 MG1655, this strain was really going down a lot, even when it was expressing a little bit of GFP. Something interesting to point out is that although this bacteria was expressing much lower amount of GFP, the fluorescence, the specific fluorescence was higher. So probably this strain is producing a lot of GFP, but not of high quality. This strain is producing less GFP, but most of them is active. Well, with this, I hope we can just familiarize a bit more with the burden that is due to the recombinant protein expression, and that each strain of E. coli can give you a very different profile of production. Our general aims are to produce enzymes in a way that we can share them easily, and aiming to have self-sufficiency in Latin America, and of course in many other regions. I am from Latin America, so this is a general aim for us. And we also want to develop our biosensors so that we can check safety of water anywhere. And for this, the signal, we have these circuits sensing metals, but the signal, if it is very high, then the bacteria doesn't grow so well, then the sensitivity is not so good, and we have a lot of false negatives. So when I wanted to have the BL21 DE3 producing T7 polymerase as a way to increment the signal of metal biosensors, or maybe by any other thing, so I can induce the expression of recombinant protein by adding mercury with this metal biosensor. Why not try another circuit, something a bit more friendly than mercury, and maybe less expensive than IPTG, and express a lot of other protein? And using T7 RNA polymerase for expanding, for incrementing the signal is a great idea. So when I shared in the WhatsApp group of many recloned friends, what can we do if I want to do something else with T7 RNA polymerase in a improved BL21 strain? So I was thinking of using Qmate, is cumatic acid or Qmate, as an inducer. And Cesar from Chile asks if this inducer is stable. It's actually very stable, and it's less expensive than IPTG. Jenny is pointing out that we are relying in DE3 this is a prophage from the 1980s. So why don't we improve this with many other characteristics that we would like to? Fernand pointed out that for T7 induction, the balance of the expression of lac repressor is critical, and that is what we saw in the previous slides as well. So other ideas, maybe expressing lysozyme to have a self-lysis, can also help, but we can add them up one by one. And even T7 RNA polymerase can be thermostable, so maybe this is an advantage for free cell systems in a following step. So a little bit of history. To improve BL21 is actually a very ambitious aim. It is quite old, 1986. William Studer and Barbara Moffat presented this series of clones, and the DE3 is this one. where they incorporated a mutated promoter controlling the T7 RNA polymerase gene. Can we make this strain better? So what do we want? It has to be stable. So the system of expression should be in the chromosome, not only in plasmids. It should have very low baseline expression. And we should be able to, little by little, increment that expression from 0 to a lot. For many reasons, people who try to study different difficult proteins, they want expression to be very low, not only because it causes burden to the host bacteria, but also because they many times are toxic for the bacteria. So if you express it too much, at the end you have nothing because it kills the bacteria. So other requirements, other kind of requirements are that the inducer has to be low cost and the inducer has to be stable. Also that we can share it easily, well-documented, fully annotated sequence. It took me a while to document everything that happened with BL21 and DE3. It is published and it is present in public databases, but it's not fully annotated. And of course, we want to achieve with these tools self-sufficiency. I will go further with this. Okay. So tunability is a very important and useful feature to avoid too much burden for the bacteria and to also build new routes and to test new transcription factors. You want to test them little by little. So this is something else that is interesting for our group as well. There are other people who have produced very tunable sequence. So IPTG is not bad. If you add very little IPTG, it will produce lower amount of protein in the DE3 system. But if you start to add a little bit more and more, then you have an abrupt increase up to the maximum expression. So it's not really linear. Arabinose is much more linear, but it normally doesn't express a high amount. There is a recent publication of a marionette system where they introduced 12 different recombinant heterologous transcription factors. And that is interesting, but it's not tuned for high expression. Ribotide is something very interesting that a group did for tuning down the expression of T7 RNA polymerase. I will show that. And also the expression of this bacteriophage protein that inhibits E. coli RNA polymerase. Then you can have more of the machinery devoted to your heterologous protein. But that is not necessarily very tunable. And then this transcription factor, depending on humate, I will talk a little bit more about it. So, what happens with BL21? There are DE3, there are some precedents of problems about it. It expresses the T7 RNA polymerase in such an amount that it is very common to see mutations in the promoter. So in the DE3 insert that is in the chromosome since the 1986 work of Barbara Moffat, William Studier, they introduced this very, very powerful promoter to produce a lot of T7 RNA polymerase. But it is very common to have a recombination between the native lac promoter and this mutated strong promoter. And with this homologous recombination, you have now a weak promoter producing T7 RNA polymerase. And this event apparently is happening in almost every experiment using BL21 DE3. Inadvertently, it goes to a lower expression very quickly. And sometimes that lower expression is what you want, because it happened only after this mutation that you have a good amount of protein, because it will be expressed in a more in a slower way. And in other occasions, you don't want it, because it will go, your production will go down spontaneously. And what you do normally is to refresh the plasmid. You take the plasmid out and put it in a fresh BL21, so that this recombination will again happen, but it will take some time to happen.
Sorry, Danielle, can I just stop you
Yes.
briefly? It seems like a few people are having problems seeing your slides. You see the black screen, or is on the WhatsApp slide? Were you moving on with your slides, or?
I was moving on slides, yes.
Yeah, do you mind just doing a quick refresh, stop screen sharing? Because I think that might help.
Oh, my screen is paused. I
Yeah,
just received a. That's a pity. My screen was paused. I don't know since when. So Zoom changed a few features recently, and I'm not aware of that.
No worries, just
So
take your time, but sorry about that again.
I wonder from where it was paused.
So at least on my screen, I saw up to the WhatsApp slide conversation.
Ah, okay. From here.
So,
good.
And then I think it's just the next, yeah.
Okay.
From here.
So I was just citing the work of William Studier and Barbara Moffat, that this is the DE3, how they published in 1986. These are a list of requirements, and how the tunability is very important, some precedents of tunable circuits. And the DE3, as we know it, has this very strong promoter commanding the T7 RNA polymerase expression. But what happens inadvertently in all our laboratories is that the native promoter, the native LAC promoter, recombines and substitutes the mutated LAC UV5 promoter. So this lowers the expression of RNA polymerase, and this decreases your production. And sometimes is even better, because slow production will, at the end, make more protein for you. But can you imagine that we are expecting a spontaneous mutation to happen, so that will improve your production? That is a little bit odd, to say the least. And actually, point mutations weakening the LAC UV promoter are very frequent, spontaneous, and some of those were spontaneous in a study, and then they are fixed in the, for example, C41 strain. C41 strain is a BL21 DE3 that has the LAC promoter mutated, so that it produces a lower amount of T7 RNA polymerase. This is suggested, advised as a strain very useful for toxic proteins, so because it will produce a lower amount so that the bacteria can sustain it. Other very tunable system was published in 2018, and it was called the ribotide system. So in the BL21 DE3, they added a system, a ribo switch. So they have the DE3 system normal, as with the IPTG inducible operator here, but this controls the T7 RNA polymerase, as we are used to that. But on top of that, they introduced this ribo switch. So the messenger RNA will not be translated unless you put a second inducer. This inducer will change the structure of the ribo switch, and that will allow the expression of your gene of interest. Unfortunately, this ribo switch depends on this inducer, the PPDA. I was trying to find it, and I only find one Chinese enterprise selling it. And if you are familiar with the logistics of your institution, wherever you are, it is very difficult to have a new supplier. And so probably if I want to buy this from my institution in Peru, it will take me more than 6 months. And I'm not exaggerating, because they will evaluate the supplier, and the supplier will evaluate our credit, and then it has to be transported. And it is tough to get a new molecule to your lab if it is very exquisitely produced by only a few enterprises. So other initiatives are probably more attractive. This study by Du in 2019 did something very similar to what I propose here. They grabbed the BL21 DE3 and replaced the lac promoter that is regulated by IPTG or lactose, or allolactose actually. And they introduced the arabinose promoter, the parabat, or the rhamnose induced promoter on the tetracycline promoter. So this is the production of GFP when it is not induced and induced, production of fluorescence, not induced and then induced with IPTG, or with their new strain with arabinose. Note that the baseline is very low, but the production is also not very high. With rhamnose, it was much better. The baseline is low, and the production is very high. And with tetracycline, the baseline is significant, but the production is very, very good. So these are already improvements over the BL21 DE3. Just to add something fun, I find this very fun. I didn't know about it. I read it recently. There are versions of T7 RNA polymerase. This enzyme is so great at transcribing, and it's very useful for many things. So people have been introducing mutations to it. And in this study, Meyer in 2014, they produced 7 different, excuse me, not 7, only 5 different T7 mutants that are specific for different promoters. So this mutant will transcribe for a new promoter, a new T7 promoter, because for the original promoter, it will not bind, it will not produce, well, much less than 100th than for the mutant. And for this one, for all these mutants, they have these very nice new specificities. So we could have different T7 polymerases for different genes, and each one induced by a different circuit. We can have these 3 different circuits for 3 different T7 polymerases, and then the one I'm producing here to increment our set of tools. Okay, but arabinose, rhamnose, tetracycline, IPTG, all these inducers have some advantages, some disadvantages. Let's see. I was looking for the costs in just a price list in catalogs. IPTG for 5 grams will go between 350 to 500, and arabinose, rhamnose, cumate, naringenin, or tetracycline. Of course, 5 grams of tetracycline is extremely expensive, and nobody buys 5 grams because you don't need so much to induce. So in this table at the end, this is what I want to show. What would cost you to induce 1 liter of culture for maximum expression using any one of these inducers with the best system available right now? Then it came out to be $5 euros or 17 euros, depending on your supplier, if you use IPTG for 1 liter of culture. This is quite expensive. Other inducers are much less expensive. Rhamnose was excellent here in this experiment, right? Very low baseline, very high expression, but the inducer is more or less expensive, 12 euros for only 1 liter. That is when cumate is really is shining as a very, very nice alternative with only 22 cents up to €1 per liter. Tetracycline is reasonable. but in this system, at least, it has a high background, a high baseline. So this QMate inducer, what do we know about this? We have been using QMate in my lab. We produced this system in a plasmid, actually in 2 plasmids, well, actually in 4 plasmids. A low copy plasmid with a strong promoter, low copy plasmid with a weak promoter, high copy plasmid with a weak promoter, and high copy plasmid with a weak promoter. And this is the circuit. You have a constitutive promoter producing the repressor, and the repressor, well, it represses the expression of your gene of interest. In the presence of the inducer, you de-repress your gene of interest. And well, we have produced these different versions with a weak promoter and a strong promoter for our gene of interest. These are our results. This is just the growth of bacteria so that we know if it is toxic or not toxic, the production of the protein and the amount of the inducer. And this is the normalized amount of fluorescence per amount of cells. And you can see with the strong promoter, PN25, we have baseline expression. This is the baseline expression of GFP is being produced even without inducer. But with tiny amounts of inducer, with up to 40 micromolar, we increase a lot the production of the gene of interest. With the medium-low promoter, a weaker promoter, we have no background, no detectable background at all. So our plasmid would produce 0 gene of interest in the absence of QMate. Then we have to add higher amounts of QMate to induce up to 1 millimolar of inducer to produce a high amount of GFP. So you see, with a weak promoter, we have it very tightly controlled, but we need a lot of inducer. And with a strong promoter, we have baseline. We have baseline expression, which some leaking, which we don't want, but we can save a lot of money with the inducer. Well, this is a little bit more of the same thing, but with another strain. We tried this with DH5 alpha, with K12 MG1655, and also with BLR. BLR, as BL21, has these proteases knocked out, so it is best suited for protein expression. You see this curve, we are very proud of it, is very, very tunable. And it is when you have this very tunable system that we came out with the idea we should use QMate to produce T7 RNA polymerase, because we can finely tune it. Well, with BL21 DE3 also. And of course, when you have what you think is a good idea, somebody else has done it before. And Marionette is a system that was published in 2019, where they introduced 12 different transcription factors into E. coli. And our repressor QMate, or CMAR is the repressor induced by QMate, is one of these 12 that they introduced into E. coli. This is a huge work that they have done. The Voigt lab, they have done evolution, not of the transcription factor. The results that I showed from my laboratory were done with the transcription factor that is native from Pseudomonas putida. In Voigt lab, they use this promoter and the transcription factor to evolve it to a much tighter and broader spectrum transcription factor. And so if it is better, let's use it. This is a little bit of data from the publication where they have tried different promoters for the production of the repressor, and also different settings, configurations of the operator, the binding site of the repressor. And with the native transcription factor, they have this curve of induction, and with a mutated one, they improved it 10 times almost. So they have a broader spectrum with no baseline and a very high amount of expression when they want it. So they produce this mutated transcription factor and the best configuration of the operator as a part of the promoter in two copies. So that there are two binding sites of the repressor. Well, let's copy it, right? And what I intend to do is to bring this transcription factor to be produced in E. coli constitutively in the chromosome. And this repressor in the chromosome will repress the production of T7 RNA polymerase. For this, the strategy is recombinering. So we need homologous sections flanking the whole construct. And this is the construct that I have been making in the last 3 months. Well, 2 months. So we need 50 base pairs of, excuse me, there is a mistake here. It should say integrase. And I'll go back again. Can you see my scheme of blocks? Yes. So for recombinering, for introducing this construct inside the chromosome of E. coli, we need 50 base pairs of homologous sequence on the left flank and 50 base pairs of homologous sequences at the right flank. If we want to introduce this into BL21 DE3, then, well, I had to study the sequence of BL21 DE3, and there are a lot of sequences from the prophage. So leftovers from the prophage that Studier and Moffat used to introduce the DE3 insert. There is a lot of prophage of bacteriophage proteins there, probably not expressed because they would have the promoter from the bacteriophage. But when you produce T7 RNA polymerase, probably they are going to be expressed. Well, it works. BL21 works very well. So those proteins are still there. It's not clean, but it is there. But well, one of the proteins that is partially there is a pseudogen already, is not complete, is an integrase from the bacteriophage. So I chose these bases, these sequences from the integrase as the left flank, and in the right flank, sequences of the T7 RNA polymerase. So everything in between will be our circuit commanding the expression of T7 RNA polymerase. I can go to make a zoom in into this design. So in this part, in my left side of the construct, is the constitutive expression of QMate repressor, QMate dependent repressor. This sequence has been taken from the marionette system. This is the, excuse me. At this side, you have the starting of the amino acid sequence of the SIM-R repressor, already with the 2 mutations that they introduced by directed evolution. And here, the promoter, constitutive promoter for E. coli. This is a synthetic promoter, and they have tuned this promoter and the RBS for repressing a plasmid. So I have not changed this for now, because the goal of this repressor is going to repress a chromosomal single copy gene. So I flanked this with restriction enzymes so that we can change the RBS or the promoter eventually. And then I will zoom into the right side of my construct, the operator. Here, here we have the -10, -35 of the promoter. This is a synthetic constitutive promoter that will become inducible because it is repressed by the binding of SIM-R in this operator, and this operator, the repressor will bind. and they will allow, when they are de-repressed, they will allow the expression of T7 RNA polymerase. So in this flank, I have the homology site for recombination with the DE3 insert. This is the beginning of the T7 RNA polymerase. What happens if we produce too much RNA polymerase? Well, you can add a lower amount of QMate. But well, we have to characterize the whole slope of induction. And if it happens that that we're expressing it too much and the repressor is not enough, we can tune it down by changing this RBS. For that, I flanked it with restriction enzymes so that I have a set of 4 different RBSs for higher or lower expression. This is it. I want to thank especially to Angeline Rebull, who is accompanying us right now, because she helped me with all the work in the laboratory and with the design of this construct. Damián is the host in my stay here in CABD, the Centro Andaluz de Biología del Desarrollo. I am here for almost 3 months, thanks to funds from Refract Network. It's a network for the study of tandem repeats, proteins with tandem repeats. The expression of these proteins is quite difficult, so they will benefit with the tunability of this new system. Nicolas and Dalin are students in my laboratory, but no longer students. They have graduated already. Dalin has done many experiments with the QMate, and Nicolas has done the experiments with the burden, quantify the burden with the GFP. Further to this development of our QMate inducible T7 RNA polymerase, I would like to know if you have other requirements. I have produced a poll. 10 people have replied to the poll. Thank you for that. I would like to know more about your opinions.
Thank you very much, Daniel, for giving the overview of the project in whole. And I've just posted in chat again your survey if anyone has any ideas afterwards that they want to bring up to Danielle. But I'm just going to open the floor now to questions. Does anyone have any questions, any suggestions that they want to voice now? Whilst everyone is thinking, then I guess I will ask a quick. Oh, sorry. I was just checking the chat. Joseph, you were saying you're planning now to switch from IPTG?
Okay. Oh, yes. Yes. Well, so the T7 RNA polymerase. Bye, Angie. Thank you very much. Angie is living right now, Sevilla. Okay. Bye. Thanks a lot. Thanks a lot. Okay. The BL21 DE3 right now expresses T7 RNA polymerase only when induced with IPTG, supposedly. It actually has quite a significant leak. Even small amounts of T7 RNA polymerase may cause a lot of expression of your T7 gene of interest. The thing is to change the lac operator. Instead of being repressed by lac repressor is native of E. coli, so it interferes with the native regulations. Instead of using lac repressor, we're going to use SIM-R, the repressor induced by QMate that is native of Pseudomonas. It is from the family of tetracycline repressors. It doesn't have a homologous in E. coli, so it would not interfere with E. coli native regulation. And as I have shown in our lab, and other people have also shown, QMate repressor can be very tight. So it's better than lac repressor in the sense that it will not allow, if expressed in the correct amount, it will not allow baseline expression of the gene of interest. expressed. It is less leaky. So once it is put there, you don't need IPTG. You only use QMate. And QMate is less expensive, probably more stable. I would say probably because I don't have the data of shelf life, but it is stored in ethanol. And yeah, it is tunable, and so the linear range is also very, very broad. It is not abrupt as in IPTG, where you add a little bit and then you have a lot. With QMate, you really have a broad linear range.
Can you hear me?
Yes, Joseph.
Okay. What I was saying that IPTG-induced E. coli strains are more common than the one that you are creating. So I can help with one idea I've read about nearly 3 or 4 months ago that uses IPTG, but in a more economic way by not putting the whole amount of IPTG to induce the production of enzymes or proteins, but putting it in a graded order in order to produce the right amounts with the minimum concentrations and amount of IPTG. And they, as they claim, this research that I've heard, that they can reduce the cost by double. So I think before using the cumatic acid inducer, I think this could be another replacement for labs that can use only IPTG, or until this strain becomes more popular or used by many. Thank you.
Yes, I think it's useful, and officially we should use 1 millimolar or 0.5 millimolar of IPTG, but it's quite common that 0.1 is enough, and even better sometimes. So yeah, I agree with that idea. And normally we do that in small cultures, and then you go to a bigger culture, hoping that it will keep that that feature. It works. I would like to add one thing. The PET plasmids, they have the lac operator. I intend to, well, and they have the T7 promoter. So this strain would be able to express from PET plasmids with no problem. But the PET also have the lac operator. If you don't overexpress lac repressor, but actually the PET overexpress lac repressor. So the best would be to have a set of plasmids that don't have lac operator and don't overexpress lac repressor. But if you want a tunable system, you can use your PET, and you have 2 inducers. You have the QMate, but it still is repressed by the lac operator in the plasmid. So you can add or not add IPTG to further tune. Then it becomes more expensive, but you have 2 inducers to have a very, very tunable system in case you want an exquisite curve of induction. But I intend also to produce a PET28 that is mutated without the lac operator and without the expression of lac repressor. Lac repressor is messing up with native regulations of E. coli. Thank you for your idea, and I would like to get rid of IPTG because it is expensive and not so stable. But would this new strain of E. coli be available? Yes, totally. I will put it available as soon as possible. It might be good to tell us at what point this construct is. Yesterday, I had a band, the correct band of the whole construct, but without the operator. I have to add the operator as a single final step because it has a repeated sequence, so that I could not just send it to sequence as a G block because of this repeat tandem of the operator site. So I need to cut with KPN and BAM and add with a traditional ligation just one side, and then put it in the chromosome. I intend to put it in BL21 DE3, but also in a BL21 DE3 6-pack. The 6-pack is a BL21 DE3 strain developed in Hungary. I am sorry not to remember the name of the author, that expresses the rare tRNAs. And expressing the tRNAs under the control of ribosomal promoters. So that strain is fantastic because you don't need to optimize your genes. And even more, if you want a cell-free system that doesn't have to renew tRNAs, you can use a heterologous gene that has mainly rare codons, so that in your cell-free system you have still a lot of abundant tRNAs that are rare. Well, those are subtleties that improve further improvements. So I would really like to hear for more ideas. Do you synthetically design your promoters? I don't. I copy synthetic promoters. I haven't designed synthetic promoters myself. But synthetic promoters are out there, and they have certain different ranges of expression. So I picked from those. Also RBS. How would the new system compare to optoinduction systems like PDON, for instance? I don't know it. I don't know PDON. So Adrian is introducing this idea that I don't know. But optoinduction means that you have to induce it with a certain wavelength that demands a certain setting. Blue light, okay. I don't know. I don't know. Maybe it's a good idea too. I haven't considered it, and I don't know it. I guess buying the blue light is less expensive than renewing all the time the inducer. But I don't know how tunable that is. I would like to know more about that system. Then you need a darkroom when it is growing, and then you, well, it sounds fantastic.
Yeah, you just cover it with the box. That's it.
Right. That sounds very, very interesting. I'm eager to know more about it. Have you ever tried autoinduction? I haven't. I know that it can work very well. And autoinduction, I think, can be great for inducing big amounts of proteins. I don't think it's tunable. So if you want to induce tiny amounts or find how much protein is sustainable by your bacteria in the case of producing membrane proteins or toxic proteins, I don't think autoinduction will help you. But for overexpression of a lot of protein, autoinduction, as I have read, is a great system. It's a great idea. I haven't tried it myself. IPTG is considered carcinogen. How about a new one? Okay, I will check on that. I don't know. I don't know. It is digested normally by Pseudomonas putida. So that's why it is an inducer, because it induces an operon for digestion of this. I don't know how. I have not drunk it yet, but we have not been careful with it, and it has, the MSDS doesn't have any special warning. But I think to check. I didn't know IPTG was considered a carcinogen.
Great. I think that was all the questions in the chat. Are there any more questions from the rest of the audience? Yeah.
Can we share again the link of the poll?
Yes, I will. Give me 1 second.
And I actually prepared an Excel file with the replies that I had at that moment, and I lost it. So I have to do it again. And I was pleased to see that many people have similar problems to saying that IPTG is not the best option for them. Also that that they want to keep using the same plasmids, that it's a huge advantage. And that mainly tunability is the first thing to try. Going to low temperatures is one way to tune the expression down, but also it expresses some chaperonins, right? Some chaperones, some special holding, aiding proteins. So it's something else. So low temperature is a great strategy. I love to work on an auto everything strain like this, but with some improvements. Improve release of okay.
It basically does autolysis of the cells, and then it expresses a nuclease to destroy the DNA and RNA. But they're all auto-induced as the culture progresses. So you just freeze thaw the cells, and they lyse, and all the DNA is gone. Minimal hands-on time.
I am very scared about having an auto-induced lysis, because then you will have to be very strict with the time control. If you leave it lysed for too long, once you lyse, it is, I mean, you know, when purifying proteins, once you lyse, you cannot freeze it. You have to go all the way until purification.
Yeah,
But
so it doesn't,
an induced
lyse
lysis would be great.
Yeah, so it induces the expression of the lysis genes, but it doesn't, you have to freeze thaw it in order to release them. So they accumulate through autoinduction, but then you choose the point at which you freeze thaw, and the lysins are distributed.
Yeah. I think it would be great to add the autolysis because sonication or French press or beads, they're not cheap and they are tricky. They can be different for different strains. Well, E. coli is E. coli, but yeah, yes. And sometimes they don't lyse well. It has happened in my lab that the probe of the sonicator wasn't very tight. And the person who was producing protein at that moment didn't know the sound of the right of the right setting, because you know when it's not working. But if you don't have experience, you don't know. So why not something more simple? Definitely. So it's autolysis, and autolysis helps a lot. And there are some systems for autolysis that are very, very efficacious performant. So, yeah. After I become an expert in recombinering, we should continue adding things to the strain. I'm happy to have this construct now, and I will consider it as a shuttle for different sensors. We carefully added restriction sites, especially of enzymes that are not particularly expensive, because enzymes can be 20 times cheaper or more expensive in New England biolabs. And they sell you more or less by the same price, but one has 20 units and the other one has 10,000 units. So, so the prices are very, very different. So we chose inexpensive ones. And with this construct, we can put different circuits. If QMate is not good, we can use another one. But QMate is good. As a plasmid, is fantastic. So I am very confident of that, on that.
Well, I'm definitely looking forward to see the product of this when you manage to get it all packaged within the BL21 strain. Share that with the rest of us here, so we can also test it out for you as well and give you feedback, hopefully. I am conscious we're a little bit over time, just because it seems like everyone's been having some good suggestions and good questions coming forward to you, Danielle. But thank you again for presenting to us. I'll put the survey in chat again, but also feel free to drop onto the Recon forum so we can continue discussions about this and hopefully help Danielle build an even better strain.
Thank you very much, and I hope it becomes popular.
Yes. First we can share it out to everyone, and then we'll go from there.
The first how you say that? The first one who asks for the strain will receive QMate for free.
Incentive. Great. Well, on that note, thank you again, Danielle. Thank you everyone for joining us today. And hope to see you again at the next meeting.
Thank you very much.
Bye.
Thanks,
Thank you.
everyone. I know.
Bye, guys.
Thank you very
Bye.
much, everyone. Thank you.